My name is Jack Gilbert. I'm the editor-in-chief of mSystems. We wanted to create a series of virtual mini-conferences that highlight some of the best research in the field of systems microbiology. This inaugural conference is focused on diversity, equity, and inclusion in microbiology, which highlights scientists who perform impactful research in systems microbiology and have a history of substantial contributions to DEI efforts. Our three exciting speakers today, Rosie Alegado, Michael Johnson, and Kat Milligan-Meyer, discuss their most recent research, as well as lessons learned and approaches to improve DEI in their field. One of the reasons for these meetings was to ensure that attendees get an opportunity to interact with the speakers, both through our Q&A function, as well as through small group sessions with each speaker at the end. At the end of the three lectures, we'll also have a 30-minute panel discussion with all speakers and participants. So be sure to hang around for that exciting opportunity to hear the speakers interact. I want to remind all attendees to enter your questions in the Q&A feature. And a recording and transcript of this mini conference will be sent to all attendees a few days from now. Thank you, and let's get on with the conference.
My name is Yotam Suetz. I'm an assistant professor at Johns Hopkins Bloomberg School of Public Health, Department of Molecular Microbiology and Immunology. And it is my pleasure to introduce our first speaker for today, Dr. Rosie Alegado. Dr. Alegado is an associate professor of oceanography at the University of Hawaii, Manoa, and was also the first native Hawaiian that was tenured in her department. Her group studies bacterial interactions across biological scales, from host microbe interactions, where Dr. Alegado's research significantly contributed to our understanding of Choanoflagellates as model organisms, and to microbial ecology of coastal estuarine systems and the effect of bacteria on food webs in the context of climate change. Dr. Alegado combines and leverages her research for the benefit and protection of the islands and native people, and specifically in the face of climate change. She is also the director of the Sea Grant Center for Integrated Knowledge Systems at UH Manoa, which calls to implement and perpetuate the cultural knowledge of Hawaii and the Pacific Islands to current science and research. So I'm personally very excited to hear more about this synergy between research and activism. And without further ado, Dr. Alegado, thank you for joining us today, and the stage is yours.
Hello and aloha everybody. My name is Rosie Alegado. I am Native Hawaiian, or Kanaka Oiwi, and I'm an associate professor of oceanography at the Center for Microbial Oceanography Research and Education at the University of Hawaii at Manoa. Today I'm going to be talking about evolution, ecology, and equity, insights from community-embedded research, microbiology in indigenous spaces. I want to start by thanking the organizers of the mSystems Thinking Series for inviting me to share my work with you today. If you need to reach me, you can contact me via my Twitter handle, which is @Algoraphagus. I study a lot of things, and a lot of it is driven by the interests of my students. But if I had to put a big tent around everything that I do, it would be that I love everything that has to do with microbes. I'm particularly interested in how microbes affect their environment, and in turn, how the environment affect the evolution of microbes, their biology, the compounds that they produce, the signals that they transmit and release into the environment, as well as the relationships that they form with different entities. And this is just a snapshot of all of the projects that are currently ongoing in my lab. In particular, like I said, I study everything from the compounds that microbes produce to communicate or that are picked up and interpreted by other organisms, to the cellular structure, to organs, to host microbe interactions, and even to very large scale systems. So my work goes across a very large spatial and sometimes temporal scale. But today, for the talk, I'd like to talk to you about my work in a coastal estuary. I'm really interested, as I said, about how microbes influence in the environment. And in the context of this coastal estuary, I'm particularly interested in how disturbance and how storm frequency influence microbial community assemblages. So as I said, I'm really interested in understanding how microbes are influenced by the environment. And one particular facet of that is disturbance caused by storms. Climate change is predicted to increase the frequency of storms. And based on this model, we can see that the global annual frequency of tropical cyclones has increased quite a bit over the past 50 years and is predicted to continue to increase. In the map on the right, you can see the particular area of the world And in this case, the Pacific, where I'm from, is centered. And you can see that the tracks of the density of storms has also altered a lot. And that in particular, Hawaii, which is right in the middle of the Northern Pacific, is thought to be a place where storms will definitely increase. So I'm interested in how storm frequency will alter microbial community assemblages in the coast in these regions of Hawaii. The reason why I think my study are so important is that there have been a number of studies of estuarine bacterial plankton assemblages in response to physical forcing, but as you can see, they've either been in temperate regions or they've only focused on, with good reason, very specific groups, usually having to do with pathogens. It's very rare, and you can see I've color-coded it here, ones that have looked at the total bacterial plankton community. And so my work in Hawaii really kind of helps to provide a really necessary added-on piece to that. And in particular, I do my work in partnership with a Native Hawaiian organization called Paipai o He'eia. Let me explain where my work happens. So my work happens on the island of Oahu. If you look at the left panel on the inset, we actually do our work on the windward side of the island in this area called Kaneohe Bay. And within Kaneohe Bay, there's a watershed called He'eia. And this is the image that I'm showing you right here. So He'eia is a very beautiful tropical place, and it is composed of 3 little inlets, Ha'iku and Ioleka'a, that go down into the He'eia watershed. And the streams pass through the wetland and then end up in the fish pond. And what happens is, in the image on the right, the white circles represent all of the sites where we take our samples. And what happens and the way that the fish pond works is it's 800 years old, and it combines and mixes in this constrained area because Native Hawaiians built a fish pond wall. They also built these gates so that they could have exchange and mixing of fresh and brackish water. Within this fish pond, this exchange of brackish water, this mixing, which contains a lot of nutrients, stimulates photosynthesis and therefore the replication of primary producers or phytoplankton. However, over time, there's been development along one side of the fish pond. There have been storms that have impacted the way it's functioned. There have been a lot of sediments. And so the functioning of the fish pond has altered such that we now have identified that within the fish pond that was once very well mixed, there are actually regions where the water quality or biogeochemical characteristics seem to be very different. And we think that that also means that the microbial communities in these areas are also different. This provides a really interesting system for which we can look at how human change as well as natural climate forcings alter how this community is structured. So another way to put it is we have marine sources and we have terrestrial sources all coming together in this fish pond and mixing. And the image in the middle kind of shows the exchange. My lab uses both biogeochemistry as well as microbiology to look at and understand how the cycling of nutrients in the fish pond by microbes occurs. And ultimately, that's really important for the Native Hawaiian practitioners of this pond because they're trying to grow fish to feed the community. And so, as I said, storms or floods or rain events, anything that is a disturbance or a forcing, will either alter the influence of either the marine source or the terrestrial source. And that will then have an impact on the biogeochemistry, which will in turn select or have impacts on the microbes. So one thing that could happen is if you have a storm that brings a lot of fresh water, it can introduce new species. And the way that we can tell that is that we physically collect the water, we filter it on these filters, and you can see sometimes if there's a storm, there's a lot of dirt in the water and we can pick that up. And then we isolate the genomic DNA, usually the 16S or 18S, which represents prokaryotes or eukaryotes, and we amplify that and sequence it. And that tells us who's there. Our general questions are, we want to detect the baseline to understand what is the core microbiome of this place and how seasonal fluctuations affect the communities in this fish pond. Because we are working with a community partner and they're really interested in the production of fish, we want to try and understand how different levels of the food web or connect to one another, or how these trophic levels interact. And so we're able to use co-occurrence networks to see when are different kind of species more frequent and whether or not they move together or they correlate positively or inversely or negatively to one another. And finally, I'm particularly interested in looking at how pulses disturb the natural baselines. So stochastic events like storms, how they facilitate maybe new state shifts. So I'm going to talk a little bit about a storm in particular that happened in 2014 that brought really heavy rainfall. It started off as a tropical depression way out in the Eastern Pacific, and then kind of slowly got really disorganized, but dumped a whole lot of rain on the island of Oahu, and in particular at the fish pond. We were lucky to have both in situ instrumentation in the fish pond at one site, as well as my team was collecting discrete bottle measurements to look at the physical parameters. So we were able to capture that this was a very strong precipitation event. That's the third line at the bottom, that there was probably flooding that happened. You can see the stream came up very much so, that the salinity of the fish pond, or how salty it was, went all the way down to 0, so it became fresh, and it took over 17 days to recover. And that with it brought a whole lot of turbidity, or a lot of sediment, into the fish pond, and that these, of course, also must have changed the bacterial community. So we saw that the storm increased bacterioplankton community diversity, although it did return back to the level of diversity that was before. But let's try and pick apart what that pulse of community diversity meant. And then secondly, if the community that did actually return to what it was, or how resilient it was. So we can see, if we look at how the different bacterial phyla relate to each other, that in fact, during the storm, which is that middle period, you do have the introduction in the relative ratios of microbial populations really did shift during the storm. And when we look at a dimensional analysis that looks at the drivers of how similar these communities are to one another and how they correlate to our biogeochemical parameters, we can really see that, as we predicted from our physical parameters, that this is the storm community, which is in red, is very, very different from the pre-storm community as well as the post-storm community. The pre-storm community is in purple, and the post-storm communities are all in blue. So you can see that you had one community, it shifted because all new things came in with silica and all of this particles. And then we're seeing that it is returning back. Now the question is, it returns back, but it doesn't go back into the exact same place. So who are in those different kind of little spots? But what we're seeing basically then is, something that's a temporary shift, and then we're coming back again. So these bacterial plankton assemblages are co-varying, and it seems like are in fact being driven by environmental conditions. And those specific environmental conditions seem to be ammonium, which is coming from the wetland, which is upstream from it, as well as silica. And because the Hawaiian Islands is volcanic, what that tells us is there's a lot of water that has been running through rocks and weathering processes that are occurring. So the bacterial plankton assemblages, what we did find when we took all of those individual species and we kind of grouped them together, I'm kind of glossing over a lot of really specific statistics, but you can ask me more in the question and answer period. Basically, what I'm showing you here is, if you group all of them and we let them cluster and sort on their own, the pre and the post samples seem to be more similar than the storm samples, meaning that the microbial groups in those communities that are driving the this cluster analysis are more similar to one another. And now I'm going to show you, kind of pull back the curtain. And so if I actually show you the operational taxonomic units, which are microbial speak for the microbial species, you can see that there are definite patterns of what looks similar and what looks different. And we can sort it both by date, and you can see that they sort together more by date than by site. And I've color coded, and just to remind you, those zones that we had previously identified in the fish pond. So what we're seeing is that the storm really comes in and it changes the character of the entire fish pond, such that they're really sorting by what happened on that day, whereas where are you in the particular part of the fish pond. In general, though, let me summarize, we do have a core microbiome or microbial community in the fish pond that remain persistent throughout. You can see that both at the top and the bottom of our heat map. But we also saw a really strong non-storm community and a storm community. So let's dig in a little bit more about what came in with the storm. But if you have really good eyesight, you can kind of see where I'm going with this. So what we saw is that the relative abundance of fecal coliforms in other words, poopy bacteria, increased during the storm, along with opportunistic pathogens also increased during the storm. And so what that means is these likely came from contamination, from wastewater, from people's houses, and that because of the increase in water, perhaps we had some breakdown in our sewage systems, or we have some sewage systems that aren't really systems, but are on site, like cesspools or septic tanks that probably need to be changed out or fixed. So our general findings were that when you have this perturbation, the storms definitely increase the pathogen load to the coastal estuaries, and we have more quantitative data to support that. We also found that the storms were definitely facilitating shifts in the microbial alternative states. And so if we were to kind of go down, you have a perturbation, the microbial community, does it stay the same or does it change? It alters. Now then the question is it resilient? Does it return to the original composition, or is now that composition altered? What we are seeing is that microbial communities are resilient to these stochastic perturbations. In other words, they're returning to the original community composition. And I only showed you 5 dates, but my team has continued to sample since 2014, and we do see that you have this state of resilience within the fish pond, and that is actually pretty positive for fish pond practitioners, particularly because they want to maintain conditions that are really good for growing fish. So I'm going to transition now to how my scientific work really blossomed into my DEI work. But before I do that, I have to thank my lab crew, who is composed of, we have like an army of undergrads. And what I really love is that my undergrads all come from the local community. And we also have an amazing field crew. We've built really strong relationships with our community partners. They're highlighted in green. Kili'i Kotubete and Mauri Gortling in particular have been so helpful in helping us sample in the pond. Our relationship with the fish pond practitioners really is what inspired the work that I'm going to talk about now. I developed a process called Kulana Noi'i, which is how you establish equitable relationships with community. Oftentimes when we go into what we call the field, it actually isn't a field with nobody in it. It's a rich community with lots and lots of people. And what we discovered over time is that our needs, sometimes when we have conflicts with community members or disagreements with places where we're trying to do research, it's because there's a lack of acknowledgement that our needs as researchers, we operate on our own time scales, right? We have graduate students, we have grants, we need to get papers. We're often operating on a 3 to 5 year cycle. Community members who are really, really engaged, particularly in culturally significant sites, their time scale is on the time scale of generations. They're here to protect their resources for themselves, for their children, for their grandchildren, on behalf of those that came before them, their ancestors. And so their needs are very different. And if we take the time to establish these relationships, we can get to this sweet spot where we can have equity and the good stuff happens, meaning that we can come together and we can leverage and create and synthesize more resources, create knowledge that is impactful to both academic researchers as well as the community as a whole. And we can both learn from each other and teach one another and really, really increase our impact. So as I said, I'm Native Hawaiian, and it's really important that when we use words from other languages, we explain what they mean to avoid cultural appropriation. So I want to explain that right now. Kulana means, and this comes from the Hawaiian dictionary, it really means your station or your rank. So one thing you have to know is, in case you're wondering as you're reading through this, why are there so many different definitions? It's because the way that Hawaiian works is that we use both literal definitions as well as deeper metaphoric definitions. It's multi-layered. So the general word for kulana is, if you're walking around, how do you carry yourself? It's your stance, your attitude, or your poise. Are you proud and standing up straight? Or are you hunched over, kind of embarrassed because you haven't done the right thing? So we use that term kulana to say this is your research stance or attitude. And noi'i, which modifies kulana, means to seek knowledge or information, to investigate or really examine. When you put those two words together, basically, what it means are, what are your research standards? How do you carry yourself in your community when you're doing research so that you can be proud of yourself and proud of who you represent? Again, this is the fish pond way before all of the restoration was done. And we really like this picture because it shows that change happens in a community and scientists can be part of that, and everybody in the community is part of that. So when we started working with the fish pond, actually a lot of people like to work there because it's a very accessible area. It has really interesting biology and geology and physics. So there were a lot of people working in the fish pond. And because of the relationship that we had with the fish pond practitioners, they came to us and explained that not all of their relationships with all of the researchers was positive and generative. And so we took that seriously. And my group, I'm also a co-faculty member in Sea Grant. I asked for help with community partners to bring together these different groups so that we could move forward and figure out a process that would have equity and so that everybody would feel fairly treated. So we had a facilitated committee. We then put together workshops with stewards, community, and researchers. And in addition, what also informed this work is we did a very extensive literature search. So I'm showing you pictures here of our actual meetings with community because it's important for you to understand that we had these meetings in place in Heia, not in a classroom or at the university. We decided it was important that we have these meetings where the community was and meet them. And what we all agreed upon and came up were 8 kulana or 8 standards with best practices. And importantly, they're not checklists, they're guiding questions. And the reason why they're guiding questions is this is intended for broad application, and you can tailor it to whatever community you work with. And it considers researcher and community perspectives, but it is very much centered in a Native Hawaiian cultural perspective because all of this research is happening in Hawaii with Native communities. So the first part, even though we came up with 8, what we realized is it actually can be subgrouped into 2 larger groups. One is called Building and Nurturing Pilina, and the word Pilina means relationship. So this is kind of like part 1, build that through respect, reciprocity. As researchers, we need to be self-aware of our capacity and who we are, who we often represent. Oftentimes, community members will see us not just as ourselves as researchers, but as representatives of a university system. And then communication. I'm not going to go into detail, but I can share the link. And then the second part called A'o Aku A'o Main, which means teaching and receiving, or love given and love received, is the much harder. Once you've established a relationship, how do you maintain that? And the 4 main kulana that we came up with is maintain a long-term focus. We should build in mechanisms of community engagement and co-review, particularly if you're working in culturally significant places that may have knowledge that is protected or sacred to those people. And then knowledge, ownership, and access should be spoken about beforehand because this community can benefit from that information, potentially in writing grants to support their educational activities or outreach. And then importantly, accountability. When a project doesn't meet these, because inevitably all relationships kind of falter at some time or another, how are we going to come back to the table and talk it out and resolve these issues? You can see some of these seem obvious, like respect or communication. Some of them are really, really difficult, and we're still grappling with these, particularly the knowledge, ownership, and access piece. We brought together, as I said, all the researchers at the university working at the fish pond. You can see there were a lot. These are just the PIs. And we spent a whole day talking it through and explaining it to them, having them work side by side with the fish pond stewards who were restoring the fish pond. And that really built strong relationships that in some cases had frayed a bit. We then felt like it was such an important thing. We brought it to the university and we worked with our Center for Teaching Excellence to create 3-part workshop series that we have since repeated several times, talking about place-based research. And so far, we've had over 70 participants from 14 different departments. And you can see in this case, because we took it to the university, we're all working together, but in a building. Importantly, our work has been recognized as being very, very critical to realizing the University of Hawaii's mission to become a Hawaiian place of learning and Indigenous serving institution. And some of our practices have been incorporated actually into research grant requirements. And I'm just kind of listing the words here from 2 of those. And we've also been able to train the researchers that have been funded by Sea Grant. And I'm showing you more pictures here of how we're taking researchers into the community, because that is such a key portion for them to realize that they are part of the community, and that is built by them working in the community. And it was also seen as really important because what we found was that researchers really want to have space to share their experience working with community and learn from one another, and have a space where they can talk about the mistakes that they've made in a place that feels safe. So I'm just going to end there and say that since we started the Kulana Nui'i process and building it up about 3 years ago, we've held about 35 workshops and professional development trainings and trained over 600 people. So we feel we're making a pretty significant impact at the University of Hawaii, and our work has been recognized at the national level by Sea Grant nationally as a best practice that should be adopted more broadly by other organizations. So with that, one of the most important aspects of Kulana Nui'i is to recognize all the people and help that we've received. And so I would like to put on here just all of the people from my lab, my graduate students, undergrads, and postdocs. The work that I shared is a manuscript in preparation, so those are all my co-authors. I have a ton of project support. And all the people who processed our water quality and our funding agencies. And again, if you want to contact me, you can reach out to me through Twitter, or you can find my work at alegadolab.org. Thank you.
My name is Hu-Tung Chu. I am an assistant professor at UC San Diego, and it is my pleasure to introduce Dr. Michael Johnson today. He is an assistant professor in the Department of Immunobiology and the Bio5 Institute at the University of Arizona. He is active in science research through events like establishing the National Summer Undergrad Research Project and the Bio5 Postdoctoral Fellowship Program through the American Society for Microbiology. And today he will be giving a talk on metals, media, and microbes matter. Welcome, Dr. Johnson.
Thank you so much for that great introduction. My lab studies metals and microbes, and I'm excited to tell you about some of the work that we're doing not only in the laboratory, but also out of the laboratory as well. So this story kind of needs to begin with the bacteria and how it actually survives in a host environment. So when that bacteria enters your body, it needs to acquire lots of things that, it can't bring a suitcase to actually bring with it. So because I work with metals, we're going to talk about metals. So, and metals are actually a cofactor or structural component of 40% of your protein. So if 40% of you doesn't work, you don't work. So the bacteria enters your body and they acquire all of these metals from within you using specialized influx systems. Things like manganese, calcium, iron, magnesium, and zinc. And the host, specifically in the phagolysosome of the macrophage, my favorite cell, by the way, it actually tries to sequester some of these particular metals. And in addition to that, it actually bombards it with metals that the bacteria, by and large, doesn't use a lot of. And you're like, wait, zinc is here and zinc is here. Well, that's because homeostasis is needed inside of the bacteria. They do need a little bit of zinc, but the excessive amount of zinc is actually quite toxic to bacteria. And of course, nothing happens in a vacuum. The bacteria actually try and efflux these particular toxic metals as well. And you're saying, wait, calcium and manganese is over here. Again, homeostasis is a thing here as far as trying to monitor the metals that it receives from its environment. But mostly, by and large, the bacteria tries to acquire iron, manganese, calcium, magnesium, and it tries to get rid of a lot of the excess copper and zinc here. And because I'm a copper guy, we're going to concentrate on copper, which is universally toxic to pathogenic organisms. So the bacteria encounters a lot of different environments, not only outside, but also inside of the host. Inside of the host, it uses all of these import and export machinery that you see listed here to try and monitor how much metal it has inside to work with a lot of the proteins and enzymes that it has there. And in the environments inside of the host, the metal content can change radically, especially during infection. As you can see, the copper concentration goes from 11 to almost 4-fold increase in the blood. We see zinc radically go up in a lot of these particular environments in the nasopharynx, brain, and lung. And in the phagolysosome, we can actually see that copper up to 500 micromolar it gets actually up to 500 micromolar inside of that phagolysosome when the phagosome fuses with the lysosome and makes that big ball of death. But again, since we're concentrating on copper, let's concentrate on this particular export system here. So the export protein for copper in this system will be COP-A for you all to know later, and we'll get into that in the talk. So why should you be a fan of copper, as much of a fan as I am? Well, we know that copper was used in the time of Persian kings for food preservation. They put their stews in the copper pot, and they knew that a lot didn't grow when it was in the copper pot. They also combined it with lye to make the Bordeaux mixture. And with the Bordeaux mixture, they sprayed that on grapes for vineyards. They also sprayed it on potatoes and other plants. So if you like wine and you like French fries or baked potatoes, think copper. Copper's your new favorite metal. Go team copper. Now, you can also use copper tools and surfaces to cut down on hospital-acquired infection up to 70% as compared to stainless steel. And again, I said that you use copper to not only kill bacteria, but also in a lot of the proteins that you use. So it's actually part of your diet here. So copper is one of those really interesting metals. It's redox active like iron is, but it's really interesting niche that your body actually uses copper to deal with pathogens or to basically have a lot of cellular processes, but bacteria don't. It's almost like what mercury would be to us, copper is to bacteria. So it's a very interesting metal in that regard that we can actually use it, but we can also use it to kill bacteria, which is what makes it an interesting metal to study to try and see if we can work with it to kill bacteria. So the bacteria we work on actually is Streptococcus pneumoniae. It's a causative agent of sepsis, meningitis, ear infectious pneumonia. It's carried asymptomatically by many in the nasopharynx. There's 100 different serotypes, and we have a vaccine against 23 of them, and that leaves 77 that aren't accounted for, and there are now unencapsulated strains that are now causing damage with Streptococcus pneumoniae. And we know that it's still a problem independent of the vaccine, or we know that number has definitely gone down from it, but accessibility matters. So we know that there are about 1 million Streptococcus pneumoniae mediated deaths per year, and about half of those are under the age of 5. So my laboratory studies how do bacteria overcome the copper stress? What can we do to take advantage of copper's toxicity as a therapeutic, and why copper is actually toxic in the first place? So going back to that export protein, and there are a number of other genes in that particular operon, one of the things that the bacteria does to overcome copper stress is have an export system for copper. And we can see if we knock out the repressor, meaning that you're making a lot more of these other proteins that help you get out the copper, it actually appears mostly like wild type, but when you knock out and this is actually a chaperone here, this COP-A gene that makes a COP-A protein, and this COP-A again, which is the exporter, when you knock those out, it's 10 times more susceptible to copper stress. We also, if we knock out that exporter, see an increased copper concentration inside of the bacteria. But none of this really matters unless in an animal model of infection, or it gives you something to go after therapeutically. So if we make a mutation to that export system, we see a great attenuation in virulence. And this is a lethal dose 100 using the wild type Tiger 4 here. We can see that it goes, the survival goes down to almost 0 versus the COP-A mutant is greatly, it basically has no death. They all survive. We see reduction in lung and blood titers. We can do, this is a pneumonia model of infection. We can do IP, IV, intranasal, otitis media. We all get the same thing, all result from this. And what this tells us is that bacterial systems used to overcome copper stress are therapeutic targets. Now, instead of going for maybe a specific therapeutic target, maybe we can go for something that's a little bit more broad. So I went to a metal meeting and met this woman. She was actually the first person I met there, named Kathy Franz, and she told me about this compound called 8-HQ, which basically it's a ionophore that synergizes with copper to kill Cryptococcus neoformis, which is what they were doing at the time. They did some great bit of chemistry to actually add a borate group to 8HQ as it's toxic in the host environment that gets cleaved by hydrogen peroxide, such as in the phagolysosome. And I said, I could not wait to get back to the laboratory to try this compound to see if it worked against Streptococcus pneumoniae. So I set it up, I was all ready to go, and it didn't work for me. And, you're not defined by your hypothesis, but just how you carry forward from your hypothesis. So let me take you through this with nothing added, and you add the 8HQ, no synergy there, a little bit of copper stress with then with the compound, no synergy, 50% inhibition at the compound, again, no synergy. But I happen to have the copper export mutant out, and I tried it with that, and all of a sudden, now I'm seeing that drastic change like they were seeing with their particular assays. So I had to think through this and try and figure out what was going on. So the two things that we came up with is, one, from the bacteria point of view, the bacteria has a rate of influx and efflux of metal. And we know that with between the Tiger4 wild type and the COP-A mutant, that the rate of efflux is drastically reduced in the copper export mutant. The other thing that we know, and I like to come up with analogies for things, is something called the Goldilocks. And we know that like Earth is in the Goldilocks area. So we know that if we have a compound that binds copper very tightly, it's actually going to end up being protective. But if we have a compound that binds quite loosely, then we know that the export system of the bacteria can just kind of deal with it, as far as sealing it away. But if we have something that's just right in the middle, that it can be an ionophore, carry copper in, and maybe that copper can be delivered to whatever protein it might be, maybe mismetallating, or that's basically disrupting the metal that it uses using copper, because copper has a very high affinity for various active sites, and metals are quite promiscuous. So that's the compound that we were trying to find, something that was in that particular Goldilocks zone. So we screened quite a few. And what's nice is we were actually able to find one, which we're calling 240W, and we're able to titrate the amount of copper and the compound. And using this concentration that's in the phagolysosome, we see a reduction of almost a little bit over 99%, and we can actually titrate the amount of other of the compound of copper here. But one thing of note here is that this was actually done in a complex media. We're giving these bacteria everything they could possibly want as far as a nutrient to grow, and that's not the way that that looking at a bacteria should always be. Sometimes the environment matters on how these bacteria grow. So we decided to take a look at minimal media, that maybe more mimic the host environment, or, that maybe mimic the environment that it might be in. And lo and behold, and let me point you to this green bar here where we see it at 10 to the 5 at 4 hours. In 3 hours, it's actually not detectable here in this minimal media. So we'll get back to the connection between the media story in a second. But first, I want to show you some great data, because again, it doesn't matter if you do this in a test tube. It matters if it works in an animal model. So or at least the model of infection that you're working with, whether it be animal or plant or what have you. But we did this bacteria because it's a pathogen to humans, and there's a nice animal model for it. We were able to drastically reduce the amount of bacteria in these 6 to 8 week old mice. You can see over the control, and also the number of bacteria in the lung as well. But because pneumococcus is also a disease of the old, we decided to try it in aged mice, or 18-month-old mice, and we were able to see an even more pronounced effect, not only at 7 hours, but also at 14 hours, at least within the lung. We tried this with MRSA, valley fever, and schistosomes in vitro, and actually found that this particular compound worked. And we're now looking at some derivatives, and the derivatives are working even better. So we're very, very excited about that particular story moving forward. And I just wanted to share that with you as a piece of science that's being done in my laboratory right now. So that's kind of hot off the presses. We're writing the paper right now. So going back to this whole media story, right? So the environment we live in is basically that media that allows us to grow. And what we do know is, from historical point of view, the whole point was to grow the bacteria, to be able to study it, not necessarily to see it in its environment, but we want to be able to get this bacteria growing so we can actually manipulate it and study it. But not all media is actually designed for universal growth. There's selective medias. There's medias that are, again, give every resource, and medias that more mimic a host environment that are a little bit more minimal. Why does that matter? Well, it matters because less than a third of Americans can name a living scientist. So what does this say about the media or the environments that we're growing the next generation of scientists? And again, which communities do you actually think this affects the most, especially when disseminating information? There's a lot of marginalized communities out there that aren't getting the kind of information that we need out there. So I have outreach in the time of pandemics, because pandemics with an S, because of the racial pandemic and COVID-19 pandemic that's been going on. Going to the nutrients, we know fewer nutrients basically produce different results. But we also know that representation matters at all levels. And we can, see that at the PI level. We can see that with Black in microbiology. We can see that in mentoring a diverse future. We can actually even see that in education efforts for the current vaccine. We know in November, the percentage of Black Americans who said they would take the vaccine was in the 40s. And now today, due to a lot of educational efforts, it's now in the 70s, people who actually have taken or will take the vaccine, which is just due to education and representation in the field. And then I'll say, you can do a lot more than you think. And yes, you out there, people say, well, who am I? No, you can do a lot more than you think. And I want to tell you about one of those particular programs of just stepping up and just trying something that you think might help your community. And when you're thinking about these things, as far as being an ally, you want to make sure that you are who you say you are when you're saying that you're an ally. So enter the National Summer Undergraduate Research Project. So a lot of people were missing out on summer research programs due to COVID, and we thought that this predominantly, well, not predominantly, but really affected underrepresented minorities who rely on these particular opportunities to go to graduate school or to pursue the next level of their education. So honest to God, this is how this happened. A few emails were actually being shared on June 2nd. You can see the date here, late at night. Well, maybe not too late for some people, but I was trying to figure out, is there a way that we could maybe gamify or provide research opportunities for the people that are missing out? So I talked to Dave Baltris, who many of you might know, and also Jennifer Gardy at ASM, to see if we can try and put something together. So we had this program idea, and we basically said, okay, let's see if we can facilitate a research program in a matter of 11 days, which sounds insane. It is insane, but there's a lot of logistics in there. What discipline? Who are the target students? The discipline was actually microbiology. What resources do we need? What information do we need? The questions just piled up and piled up and piled up. But the biggest question of all was, will there be buy-in from the scientific community? At a time where people were saying, we're allies, we're here to help you, were they going to be there when we needed them most, especially for these underserved populations, for these underrepresented minorities, for these marginalized communities? And I'm very happy to say that the community came through. They came through. There were 250 underrepresented minorities in this particular program. And 30% of them had actually missed out. So 75 of them missed out on their summer research opportunity. And we were able to provide that particular opportunity for them. We were able to supply, a lot of opportunity for people from minority serving institutions. You can see kind of the breakdown of ethnicity here as far as who we were able to serve in this particular program. And that's 250 people who were able to put a summer research opportunity on their CV that wouldn't have been able to before. So a few more stats from this particular program. When I said you all came through, 180 mentors. We had 180 mentors for the students in this particular program. And afterwards, students were overwhelmingly saying, yes, I feel more confident in STEM. I feel like I know a lot more. I feel like I should continue. They were more likely to go into STEM careers based on the survey. Also, the mentors really wanted to participate. They got something out of it as well. So if you're interested in what this all looked like, we actually had them do presentations. So on our website at nserp.org, you can actually see about over 170 presentations from these particular students. And it's actually stratified by those who we actually gave awards to, honorable mentions, and kind of the other great, awesome participations. There's even a few music videos in there that are, I think, quite outstanding in the way. We told them to get creative in how they did it. And these presentations were used by these students at national meetings, like Abercams and SACNAS, and some of them even won awards, which we're very, very proud of. But one of the things, and I have it bolded, the stat that I think stands out the most here is that while we were trying to help people who lost their opportunity to do a summer research program, we found a community that I'm naive and didn't know really existed. 33% of the students said they wouldn't have been able to travel to do a summer program in the first place. And an additional 25% said they were unsure. So independent of COVID, they wouldn't have been able to travel. And there are a number of reasons. They have to take care of their family. They can't, just move for 8 weeks like that. Health concerns, or even monetary concerns, because sometimes, getting paid by these programs, and there's a lot of programs that do pay and that works out, but still it's a financial burden to now pay rent in maybe different areas. So there's a lot of concerns with these populations. So that's the new community that we really want to serve here. And that's why NSERP will continue to progress here. So we're calling everybody, we're saying, hey, NSERPers, we're looking for mentors, people who want to partner with us to, help us fund this particular program, because we did it all from basically donations from you all last year. So we're hoping to make this year strong. We're actually hiring a program coordinator to really help us move forward in this particular project. So let me say one quick thing about what this meant to me. During a time of the racial pandemic, this was food for the soul. This was therapeutic to me. And sometimes doing that outreach is, it can be therapeutic to you, but it can also be really helpful to those particular communities. So I implore you to just maybe step outside of your comfort zone to try to do some of those things moving forward. So again, I'd like to thank the people in the laboratory who did, a lot of the great work, the collaborators that I'm working with, Dave and Jennifer Gardy, who have helped a lot with NSERP. And I'm happy to take some questions. Here's a way to connect with me. Thank you so much for the opportunity to speak to you today.
Hello, everyone. I'm Mariana Binlos, an assistant professor of pathology, microbiology, and immunology at Vanderbilt University Medical Center. It is my pleasure to introduce our next speaker, Dr. Kat Milligan-Meyer. Dr. Milligan-Meyer is an assistant professor at University of Connecticut in the Department of Molecular and Cell Biology. She is an Inupian runner, mother, microbiologist, and a beater, not necessarily in that order. She was raised in a remote community above the Arctic Circle that was about 80% Alaskan Native. And she uses an Alaskan fish, stickleback, to determine the extent that the host environment contributes to host microbe interactions. She's specifically interested in the resilience of the gut of the microbiota following disruption and how this may affect host development. She also studies how to improve STEM in graduate education, with a focus on experiences by historically excluded and underrepresented students. So take it away, Dr. Milligan-Meyer.
I'm Dr. Kat Milligan-Meyer. Inupakamiaruna, Napaktakamiaruna, I'm Inupak from the Napaktakami tribe. Kikiktagrok-miaruna, I'm going to be telling you about Kotzebue, Alaska, where I grew up, which is formerly known as Kikiktagrok. But first, I want to start with a land acknowledgement. I live and work on the land of the Mohegan, Mashantucket Pequot, Eastern Pequot, Shagatakok, Golden Hill Paugussett, Nipmuc, and Lenapes people. It's important to understand that the land that you're on, if you're in the United States, most likely was taken from a tribe that was, or a collection of tribes that were here before you were ever conceived of. These particular groups have been on this land and stewarded this land for thousands of years. Over the last 400 years, they have faced forced assimilation and genocide to have thriving communities now. And that really speaks to their resilience, which is something that I'm going to be talking about as I describe adapting an evolutionary model organism for host microbe interaction studies. But first, I'm going to start by telling you about where I came from, because this drives the research that I do. I grew up in Kikit-Tugruk, Alaska, a town of 3,000 people just above the Arctic Circle. We're the largest town in our region, and we have a mixture of Western lifestyles and traditional lifestyles. So a reflection of the Western lifestyle are the paved roads that you see and the very Western style houses. Representation of the subsistence lifestyle is the subsistence fishing net that you see on the right, which was put out by my dad in front of his house. In just 2 generations, people in my region have seen monumental changes in the way that we live and the environment around us. So this is an image of my Adato, which is my dad's dad. He's standing in front of a house that he built with logs that he took down from the Noatak River where he grew up. He's driving a dog sled team, and this was how he got around for most of his life. And he has a dog sled behind him that he built with his own hands and that he taught my dad how to build. He spoke fluent Inupiaq and had a large family. And you can see that just between the generation of my Adata and my dad, he's the baby in the picture, there were large changes here evidenced by the way that they're dressed. So although most of the people in this picture are wearing Inupiaq clothing made out of fur skins, you can see that my Anna, the woman on the right, has a cloth cover over her parka. And some of the shoes in this image are actually Western style shoes. My dad's generation underwent a huge change in the way that they lived, driven in part by comments from educators, ministers, and doctors who told us that the way that we spoke was backwards, that the information that we gained from 10,000 years living in the world's harshest area of the world was backwards and not as efficient or not as up to date as Western education and Western medicines and Western ways. And we were told that the stories that we told and the dances that we did and the traditional tattoos that we had were blasphemous and would lead us to eternal damnation. And so we changed the way that we lived, we changed our stories, we changed the ways that we collected our food to reflect this Western pressure to become more Westernized. In fact, by the time you get to my generation, I'm wearing a traditional parka, but I don't wear that in the lower 48. In this picture, I'm showing a snow machine. I did not grow up with a dog sled team, even though my dad has built a dog sled for me. I live in a house that somebody else built before I came to the lower 48, and most of my food comes from the grocery stores. What you don't see in these pictures are the large changes in the landscape that have occurred over the last 100 years or so. The climate change that we're experiencing globally has had monumental effects in the Arctic, leading to significantly higher temperatures in our region and shifts in the migration of animals that we rely on for food and the appearance of some of the berries and other plants that we collect, which changes the way that the animals who are migrating through are able to eat. And so that affects our ability to collect food that we need to survive in the Arctic. These are the types of things that we need people studying, but unfortunately, due to the way that we've been, the way that our education has been suppressed, we don't have as many people from Alaska Native backgrounds who are currently researching in STEM areas that could help the people in our region. Now, I was educated in the lower 48. I went to the University of Wisconsin-Madison, which has 40,000 students. Only 250 at the time were Alaska Native or American Indian. And for most of the time that I was on that campus, I was the only Alaska Native as far as I knew. To maintain a tie with my Indigenous self, I relied heavily on student groups such as the American Indian Science and Engineering Society and Wunkshik. These helped me maintain my Indigenous identity. And so here you can see a picture of my daughter looking up at Martine, who is wearing traditional regalia at a powwow, which is not something that my tribe practices, but just hearing the drums and being in a communal area with other Indigenous people helped me maintain a sense of my Indigenous self. I also helped, we also strived to fight for Indigenous rights. So on the picture on the left, Aaron Birdbear in the second picture, the second person in that image, led a fight to preserve the effigy mounds that were built by Indigenous people thousands of years prior. And those Indigenous people had ties to the Indigenous people who are in Wisconsin today. And those effigy mounds were supposed to be bulldozed down by developers, and we were able to save some of those effigy mounds. We also protested to implement Indigenous Peoples Day. Madison was one of the first cities in the country to establish Indigenous Peoples Day, mainly because of the work done by Wangshik and other Indigenous peoples in Madison. And then I also maintained my Indigenous self by teaching Alaska Native games. And here I'm showing how to do the stick pull with my lower 48 friends, wearing a traditional Etiquette, which I had completely stopped wearing until I started teaching these Alaska Native games again. Now I regularly wear Etiquettes to show people that Indigenous people are in this space and that we belong in this space. I'm not the first person to advocate for supporting Native groups or groups for historically excluded and underrepresented students. There's a rich volume of work out there by people who have examined how people have been able to survive academia when they come from historically excluded groups. Drs. Smith and Smith in New Zealand in particular have written books on this related to the work that they did graduating over 500 Maori people in 7 years. And some of the ways that they did this were to bring Maori students in as cohorts so that those students would not be the only in their group on those campuses. They support Maori student groups. They had faculty members for these cohorts, and they had regular gatherings for social and academic support, much like ACES and Wangshik provided for me. These are not the only answers to helping people from historically excluded and underrepresented groups thrive in academia, but these are some of the top things that academics can start doing now. Now, as I said, I was at the University of Wisconsin-Madison for both my undergraduate and my graduate work. As an undergraduate and a lab technician, I studied the genes and environments that regulate toxin production in the gram-positive bacteria Staphylococcus aureus. And as a graduate student, I studied the obligate intracellular parasite Toxoplasma gondii and characterized the genes that parasite needs to establish an infection in mouse brains. Now, these two areas of study, have major importance for people in the lower 48, but almost nobody in my region is affected by Staph aureus infections or Toxoplasma gondii. And so while I found that these were very meaningful studies and I learned a lot, these didn't help me work towards answering questions that would be important for the people back home. And so in graduate school, while I was in graduate school, people were starting to study the microbiome. And one of the things that I noticed is that a lot of people were studying the microbiome from a white-centric point of view. So the people that they would characterize the microbiomes for were people from primarily white institutions. So most of the people in the studies were white. And when they did study the microbiomes of people who are Indigenous, it was often in comparison to the microbiome of white people. And so I started thinking about how population variation in microbiota affects host development and their health, because I thought that this is something that I could actually use to help people back home. As Alaska Natives have higher incidences of obesity and heart disease and diabetes and IBD cancer, all of these are diseases that are correlated with changes in the microbiota. And so I was hoping that by studying population variation in microbial communities, that I could apply some of that knowledge to how we study the microbiome in humans. Now, I said that resilience is a common theme, and one of the things I was really interested in is this idea that Alaska Natives are resilient. This particular study was looking at the psychology of Alaska Natives and Native Americans, but I would also like to study how the microbiota of people from different populations may be resilient. Now, I don't study people. I actually use a model organism called three-spine stickleback, which has been studied for well over 100 years in evolutionary studies. And the reason for that is because there are many different populations of these fish throughout the Northern Hemisphere, each of which has evolved in a different environment. The evolution is thus thoroughly described, as is their development. They share many pathways involved with host microbe interactions in the immune response with mammals, so NIH likes that. They have natural genetic variation and natural environmental variation. These two combinations are things that we know affect the microbiome, and so we hypothesize that these different populations would have evolved different relationships with their microbes. We can get large clutches from a single cross. So if we cross one male and one female, we can get 200 eggs, roughly 200 eggs of fish that are genetically related. And the juvenile are optically transparent, which I'm showing in these two videos. So this is a fish at 3 days post-fertilization. On the right is a fish at 6 days post-fertilization. They're evolving, or they're developing inside of a chorion that is transparent. And so we can watch the development of the fish, which is also transparent inside of that chorion. And this is a live fish, which you can note by the beating heartbeat right here. This is the tail, here is the head, here are the eyes. So we can watch the internal and external development of these fish. Now that was 6 days post-fertilization. That was 3 days post-fertilization. At 6 days post-fertilization, you can see that more pigmentation is visible, but the fish is still transparent. There's significantly more development between those 3 days. And if we zoom in here, you'll see little rivers of things. Those are individual cells that are circulating on the surface of that yolk, so we can watch how individual cells are circulating throughout the host. In our lab, we are particularly interested in what happens when the diverse microbial communities in the gut, the microbiota of the gut, is disrupted, and how that affects overall development of the host. What we're particularly interested in is the resiliency of that microbial community that differs between populations. So how well do they bounce back from that disruption? But also, can we correct that disruption by adding back microbes that will help that host develop upon a normal developmental trajectory? And so we examine the microbiota between populations and their resiliency and their effects on development between populations. Now, to do this, we had to first establish that the microbiota in different populations is indeed different. So Ryan Lucas, his name is on the bottom left, went out to 4 different populations that we knew a lot about and isolated the DNA from the guts of these fish to determine whether adults harbor different microbial communities. And what he found is that an anadromous population, anadromous fish spend part of their time in the ocean and part of their time in freshwater, have a different microbiota than our freshwater populations, indicated by FW. Now, each of these dots represents an individual fish. If 2 dots are closer together, that means the microbial community in those fish are more similar to each other. And if they're further apart, they're less similar. And you can see that the rabbit slough anadromous population is different than the 3 freshwater populations, and that 2 of those freshwater populations clustered together, Westchester Lagoon and Big Lake, but Bear Paw, another freshwater population, is off by itself. And so we were really interested in how the microbes in these fish are changing the development of these fish, the developmental trajectory of these fish. And so we've isolated over 300 microbes from the guts of these fish and from Oregon and Alaska. Rachel Cramp here is showing the anaerobic microbes that we've isolated. And so we have a library of microbes that we can play with. Now, one of the things we do then is we take our fish from different populations, in this case, rabbit slough and Westchester Lagoon, which again have different microbial communities, and we sterilize the surface of those chorions that you saw in that developmental video by soaking them in PVPI and bleach. That kills all the microbes associated with that host. Then we can put them in germ-free water and raise them germ-free. We can add back conventional microbes, so conventionalize them. And here we did this at 6 days post-fertilization. And then we can add back individual microbes that we think may affect the developmental trajectory. So in this case, Vibrio. For this particular experiment, we were interested in whether the microbial community affects the developmental trajectory differently in these 2 populations, and whether exposure to the potential pathogen Vibrio would disrupt that development. And so we collected these fish early in development at 14 days post-fertilization when the fish are still transparent, and then measured their survival, immune gene expression, and development. And what Lucas and our colleagues in the lab found is that the fish had normal overall development. So the red line indicates the measurement that we took. Each dot represents an individual fish in which we measured this. The conventional fish are on the left, the germ-free fish are on the right. The control fish are indicated by the gray boxes, and the fish that were exposed to Vibrio are indicated by the white boxes. And you can see in rabbit slew that there was no difference in the overall snout vent length. And that was also true for Westchester Lagoon, which again has a different microbial community in adults. Interestingly, though, we found that the eye diameter and the swim bladder area are different in rabbit slough and Westchester Lagoon, and they respond differently to the microbial manipulation, which is interesting to us because this indicates that different developmental pathways are influenced differently by microbial manipulation in these two populations. So this is something that we published just last year, and we're hoping to follow up on in the studies that I'll do at the University of Connecticut. Now, as I said, I'm from Alaska. Alaska is an oil-rich state, and occasionally oil spills happen. The biggest one that has happened to date was the Exxon Valdez spill in 1989. Kelly Ireland, a student in my lab, grew up in Alaska as well and knew that this spill severely affected the ecosystem and that many organisms have not recovered, even 30 years later, from this monumental spill. And so she was interested in looking at the resiliency of different populations to crude oil exposure. And so in her experiments, what we did is we exposed fish at 7 days post-fertilization when they're still in that chorion, exposed them to crude oil for 7 days until they've come out of that chorion and are starting to eat, and then moved them from crude oil to clean water for 2 weeks to determine if these fish could recover from that crude oil exposure. And then we measured many of the measurements that we measured in Lucas's paper. And what she found is that the fish that were exposed to crude oil were smaller at 14 days post-fertilization than the control in both their snout vent length and their eye diameter. But at 28 days post-fertilization, they were not able to recover during that 2-week depuration. Interestingly, there was no significant effect on swim bladder length or swim bladder area. And if you recall from the work that Lucas did, the swim bladder area was affected differently than the eye diameter. Again, here we have a different response in that there's no difference in the swim bladder area between the crude oil exposed and the control fish. Now we're interested in population variation between different populations of fish. And so we took those same populations that Ryan was using and found that the Westchester Lagoon fish that were treated with oil had on average smaller snout vent length and eye diameter and swim bladder length and swim bladder area than the control. But then at 28 days, they were larger than the control, indicating that there was a time by treatment interaction in that these fish were able to recover their growth. Now, in both of these studies, we also looked at the immune response and found differential effects on the immune response, and other work in my lab has focused on behavior. And in both of these cases, we're sequencing the microbiota to see if there were changes in the microbiota in response to these microbial manipulations or ecotoxin production, ecotoxin exposure. So overall, what we found so far is that populations vary in their microbial community, populations vary in response to microbial manipulation, and we can now use this information to construct communities, because we have this library of 300 microbes, to identify mechanisms of population variation in response to microbes. And then we can also examine the evolution of the microbes and their hosts. So with that, I want to say thank you to the people who have worked in my lab, my mentors, my collaborators, and funding. And of course, thank you to mSystems for allowing me to share my story with you.
That was wonderful. I really enjoyed that. Thank you to the speakers and to our organizers. I really, I thought it was a fantastic session. What a great diversity of talks. I'm Jack Gilbert, the editor-in-chief of mSystems, or in my current incarnation, Fat Thor, as my children are calling me right now, which refers, I think, to my beard. But I thought this was a wonderful opportunity, and we have about 20 minutes now just to ask some of my questions, because no one else is about to ask me questions, which I think is great. And to have a quick chat, and then there'll be an option to spin out into some breakout rooms for those people interested in meeting some of the speakers. So I just want to start with you, Rosie. I hear you now have 18S data from the study you were talking about. Can you tell us more about what you found?
Yes, I do. Actually, it's quite interesting. A couple of things that we note is that when we looked at the alpha and beta diversity of the eukaryotes, the microeukaryotes, due to the storm, we see a different pattern. So whereas the storm, it seems, brought in new bacterial species, and you saw from those NMDS plots that the spread, what we actually saw was what looks like a flip out. So instead of bringing in diversity, we still saw a tight community that was distinct from the pre-storm communities in the eukaryote population. And we think that it may be because they're eukaryotes and they have less metabolic diversity. It may be because the microbes that remain there while there are new populations coming in, maybe they are kind of just sitting and hanging tight and have different strategies. But we find that to be very interesting. And one of the most exciting things, thank you for giving me the opportunity to use the word choanoflagellate in a talk, is that we identified choanoflagellates in this study. And so literally that was the whole reason why I wanted to start working in fish ponds, was to find Hawaiian choanoflagellates. And so it was really gratifying to be able to see that data. And now we're beginning to identify what the biogeochemical conditions are for these natural populations. And that will open up doors for us to begin experiments on looking at ecology and evolution of choanoflagellates in natural settings.
I mean, if you got a storm event coming through, right, I'd expect lots of sediment particles, blah, blah, blah. And so are these particle-associated choanoflagellates? I love that word. It is a wonderful word. Coming through, or how if there's this community that comes through only with the storms, ignoring the sewage organisms. I'm interested to understand how that happens.
Yeah, that's a great question. So I did simplify my experimental design, and we do filter at different size classes, so we can begin to discern whether the different kinds of microbial eukaryotes we see are associated with particles or are free living. I don't have that information quite at the moment because we literally got the data last week, and my first question was, do you have choanoflagellates? So, the answer to that is yes, and the answer to where they are kind of sorting in terms of the size class and what they're associating with is an unknown, as yet unanswered question. So I hope to be able to answer that soon.
I got her on mute. There we are. That you got to get your data analysis done faster. I think that's great. I was going to ask, and this is in reference to a number of the talks talking about actually engaging with K through 12 students, especially in bringing more diverse student population into the STEM environment in which we all work. I was wondering if anyone's, especially, or Rosie, it came up when you were talking about engaging with the local community, but it's a reference to you, Michael, and Kat as well. Actually bringing those the children into the academic environment and getting them excited about the realistic possibility, even when they're young, of actually being involved in that kind of community. We used to do that in London with populations that would never have even thought about the possibility of going to university, right? It just didn't exist on their radar, or their, for cultural and financial reasons. So I was wondering if that's happening in Hawaii. Is there an engagement plan for that?
Yeah, so I just have two quick points on that. One is that we have a wonderful institute called Napuna Eau. It's the Institute for Native Hawaiian Advancement. And over the past 20 years, it's educated over 16,000 Native Hawaiian children and bringing them into the STEM fields. And they have specific programs where they bring them to the university campus and engage them in programs.
And so, yeah, I myself, actually it's older than 20 years because I myself was a student in this program, and that really helped introduce me to science that was going on at the university. The other thing is that we do work with a lot of the school groups and the children who go to the fish ponds. And in that sense, we really look at as capacity sharing, as opposed to, we don't really see ourselves as kind of building up that capacity. We see the local indigenous community as having a unique and distinct set of knowledge that they can share with us that informs our work. So we think of it as capacity sharing.
I love that concept. Michael, both you and Rosie and Kat brought up the idea of financial stability to allow people to become involved. I've got 2 points on that. Number one, why I was a science question, why are we not looking at metals in the gut? Everybody on this meeting wants you to look at the heavy metal impacting upon gut microbes. Dude, seriously, missed opportunity.
I mean, I look at lung pathogens, I guess, with cocci and Streptococcus pneumoniae, but I'd be happy to, if somebody wants to, let's talk, right? I'm happy to look into some other areas, because I feel like, that is an area that if you look at the literature, maybe in the last like 4 to 5 years, now you see all these copper and gut papers, and microbiome things kind of coming up. But, I think that it's a untapped for looking at additional studies. So I'm definitely,
I got 100 questions about
that.
Like, where does the stuff get absorbed? How much copper gets into the blood? You were talking about these crazy variances in concentration and how the microbes adapt to that. And I think it's an amazing system to look at. When you, sorry, this is just for me. When you were talking about culturing the bugs and looking at rich media versus minimal media, a lot of these things you would assume are actually in a pretty rich media. I mean, when we die and we start to decompose, microbes have a field day. So I'm confused as to why they might be in a minimal media environment, even if they're on that epithelium.
So I guess it really depends on the microbe that you're looking at. So with looking at a lot of respiratory pathogens, we're looking at things that literally have to acquire all of their carbohydrates from mucus, which is kind of a difficult thing to do as you're coughing things out. Independent if you have maybe cystic fibrosis or something like that, and you can't actually push the mucus through. But a lot of these environments are really not hospitable for any type of microbe, and the microbes that have adapted to live there have done so because our bodies have kind of let them. But when things come through and say, I'm going to start screwing these up, where they basically are not really used to that type of environment. I mean, if you look at, I actually just got a review published on this. If you look at kind of these, not necessarily ancient media, but the first media that we actually try to culture bacteria in, they're just, they're full of rendered animal parts that we're just throwing everything in and try and get things to grow. We don't really know all the components in those particular systems, but we know that it made the bacteria grow. And for that particular time period, that was good enough so that they can actually look at the bacteria. However, that's not a necessarily biologically relevant system as far as those particular media. So that's maybe what I meant a little bit more as far as saying, it's like, we can say rich media, we could say minimal media. The real point is whether or not it's biologically relevant or it's relevant.
Yeah, that's super hard though. I mean, and I think Rosie you'd come at that as well when you're looking at these things floating around in the ocean, as I did back in the day. I mean, it's an incredibly complicated environment to find out what the microbe itself is sensing, right? And what it's
Absolutely.
Experiencing. I think
it's clearly metal though. It's clearly metal.
Yeah, so metal. I'll do some headbanging later. And Kat, you're dealing with these bugs actually coexisting with this well-defined substrate, which is a fish, right? I was surprised to find out that you could make these germ-free just by sterilizing the outside of the chorion. So there's no colonization of the developing embryo with inside that egg environment.
Yeah, we've been able to make germ-free fish, and so we are fairly certain that there are no microbes inside of the chorion, and we've done this numerous times. And rarely have contamination. So we're pretty sure that there aren't any microbes inside the chorion. Before I answer anything else, so I wanted to mention Alaska has RAHI and ANSEP, and these are two programs for high school students to come to, oh, and the Delegates program. So Alaska actually has 3 programs for Alaska Native high school students specifically. All of those are for Alaska Native students to come to our campuses and learn about college before they actually get there. When I was at the University of Oregon, I taught Native American students through BOGSA, and we're hoping to start something similar here at the University of Connecticut. So BOGSA, like ANSEP and RAHI, was a program to bring students to the campuses, both the University of Oregon and a community college, Lane Community College, to learn, to get adjusted to the college experience before they graduated high school. And I was actually involved in RAHI when I was a high school student, so I have to mention that because, you know, every once in a while I'm like, am I here because of RAHI? Maybe.
All right, that's really important. It brings me on to the question I wanted to ask at the beginning, which was financial compensation for engagement. You know, when we're getting out, a lot of the people that grow up in some communities just don't have the financial means to actually become involved in any voluntary activities, which, you know, unfortunately, people are looking for on you know, applications, especially into grad school or you know, undergraduate level. And so what's the framework for actually doing that? I mean, you know, are we there's federal grants out there, but that's obviously not sufficient to actually facilitate massive engagement. You know, anyone can answer.
Well, I know there's a program called DNA Day, and I know, you know, it's like you look for kind of these programs that at least go into high schools and what can you do on a national level, what can you do on a local level. Well, I think the answer on a local level is, you know, from our universities, from our institutions, we can actually do a lot. DNA Day, they give you a lot of the lectures, the materials, and you can kind of come up with things to go into high school. You can like purify DNA from your saliva, and that's like a 5-minute protocol. You can do like a genetic wheel. You can teach them about pharmacogenetics. You can teach them about you can teach them really about like any module that you want that might be relative to your particular area. And the NIH has a lot of materials through DNA Day. If you just like Google that in NIH, it'll come up. Carla Easter runs that particular program. I mean, and you'll find like all these materials. And the reason why I bring that particular thing up, I know there are a lot of formalized programs and those are fantastic, but this is more about what individuals can do. I mean, like an individual can go into a local high school. They can contact a local teacher to actually make that connection, to make connections with students. And I think that a lot of time we're just waiting around, waiting for some entity to say, okay, where can we get the funding to do this? Where can we, you know, try and help the next particular group of people? But we can also look inside and say, what can I do as an individual by myself? This is one of those things that you can actually do. It's really easy. It takes like half a day. I mean, and you make a very, very lasting impact on your particular community, wherever you are.
Well, with that in mind, yes, you can do your local groups. But what about, you know, COVID's taught us about virtual seminars and virtual interaction quite nicely. To prevent the need for people to, you know, have to travel and get travel grants to have an income there to actually, you know, have local rent, etc. Is it feasible to take some of these things like NSERP and bring them online?
So with NSERP, we're trying to be able to fund the students in the program. We did the entire operation last year for about $3,000. So we're hoping, and that was all community donations. That was literally people like, and you can look at the sponsors on our website saying, you know, we're giving you $100, we're giving you like, that's literally what funded that program. Everything else was blood, sweat, and tears, but we'd love to be able to pay the students. And I might have some news for people next week on that particular front. I'm very, very excited. I don't want to rock the boat too much, but fingers crossed, I might have some news on that next week.
So Kat, is there any opportunity to reach out virtually to the community, especially north of the Arctic Circle, when you're dealing with people in extremely remote communities, who maybe not have access to the kind of travel to these programs that they might be able to have in cities, for example?
Yeah, so there's a number of different groups who have done things like that. So up at the University of Alaska Fairbanks, Devin Drowned was able to teach a student in Utqiagvik, Barrow, which is like the northernmost city, right? How to do a sequencing using an ion, a MinION. And so that student was able to sequence microbes from the permafrost and look for antimicrobial genes in those communities. So he was able, I mean, if we're able to do that, if Devin Drown is able to do that, and Devin is an awesome professor up at UAF, you know, why can't we do that in other places? But there are other things we can do too. So like my students, obviously I started at UConn during a pandemic, and I didn't have a lot of things for my undergraduates to do right away, but I was able to find things for them to do online. So measuring fish, doing literature researches, identifying microbes in our collection that might be human pathogens that we might want to study more. So we're able to do that with the students here. Why not do that with students who live in places like Alaska, in rural Alaska? But the other thing we can do is we can bring the science to them. There are a lot of researchers who study in the Arctic Circle. Very few of them hire locals to do research. They'll hire them to show them how to get to a certain site, but they don't hire them to do the research, and they don't teach them how to do the research. Frank von Hippel has been really good at teaching people in Norton Sound how to detect perchlorate in their waters so that they can get military sites that are contaminated with perchlorate cleaned up. And he actually teaches them how to do the assays and then how to put that information together in a document that they can take to court to show that they have contamination in their areas. Those are the types of things that we can do. I think one of the things that's really important is to ask people first, what research do you need done here? For example, I'm interested in a contaminant that a new graduate student in my lab is interested in. So I shot an email, or not an email, a Facebook post out to my friends and family and said, hey, you know, what contaminants should I look at? This is something I can do with this system. And here are the other contaminants that I've looked at. I was thinking that they were going to talk about runoff from the mine site that's just north of Kotzebue, which they did talk about that. But they also said things like lead and sewage runoff and, you know, things that I wouldn't necessarily have thought of right off the top of my head. So ask local people what type of research they need done, tell them the skills that you have, and then see if you can develop research projects to help bring that science and train the people in that area to do the research. I'm an Alaska native, I can do research. That's not a barrier. You can't use it. You can't use the excuse that because they're from rural Alaska or rural anywhere or from an underprivileged area that they're not going to be able to do the research. We can, if we were all trained to do this research, other people can be trained to do the research as well.
And all that comes down, I worry that the universities especially, well, I've never received any training in how to respectfully communicate with other communities, right? And the ethical paradigm for that is woefully incomplete. So our universities, I know universities could do more, so this is a leading question, right? But in terms of actually reaching out to the white men of the world, the people like me who have had virtually every single, I've got an English accent, you know, I've got every single benefit possible in America to get to where I am. I've had to not do anything. So from that perspective, are there particular elements that could be taught and trained at the university level, and who should be paying for those? I'm hoping you're going to say the university.
Would it be okay if I kind of jumped in for a little bit? So although it would be great for the university to do that, and you know, at my university we are actively pursuing hiring not only an associate dean, but you know, also having staff, DEI staff within our department, given the current global situation and what might be tightening budgets, certainly state budgets, because I come from a state university, those prospects seem a little bit not possible at that time. However, we're not letting that stop us. One thing that I did want to talk about, it is not a microbiology effort, but URGE, which is Unlearning Racism in the Geosciences, because the geoscience has a huge diversity problem. And that was an effort funded by NSF, actually, which is a huge nationwide journal club made up of people making pods and then getting together and then reading articles to self-educate ourselves. And that's paired with wonderful interviews with the authors to understand, and then inculcated within that is we have to take action and talk to our leaders of either within our institutions or within our professional organizations of how we're going to begin to address many of these issues that you brought up. So I think those issues are amazing. In many ways, it really reminds me, I mean, it's the grassroots effort until we can get to a place where, it's an inflection point where those who are higher up in the system realize that it's to their advantage to begin to really critically take these questions seriously and address them. So yeah, I mean, that's what I would say.
Let's look forward to that bold future. I'm hoping it won't be very long. It shouldn't take long. UCSD is becoming a Hispanic serving university as well, and hopefully the budget crunches won't impede those kinds of relationships. Well, we're at the end of our discussion session. Thank you so much, guys. Everybody, we're going to be opening up 3 breakout rooms, and those 3 breakout rooms will be accepting 10 people each. And so you have an opportunity to go and meet with one of our speakers in a session that's going to be overseen by either Mariana, Jotun, or Hugh, who are our organizers, and they will oversee that. I'm going to remain here, I think. Thank you.